All right, fantastic. So with that, let's just step right into it and look at root water uptake, which is this little important thingy back here. Right? So we've looked at most of these processes in some way or the other, some in more detail than others, and some still to come. But what we have looked at in detail is this. Water ascends in this whole soil plant atmosphere continuum according to potentials. Okay? So these are total potentials. We've learned that by arbitrary choice, we define 0 megapascals to the free water without any solutes in it. And then as we go up through the soil, into the root, the stem and xylem, to the leaf surface, we become more and more negative. And then we have a big jump to the atmosphere where we're even more negative. And water, just like everything else, flows from high potentials to low potentials. But with the water potentials, that always means high is low and low is high, because we're going more negative. So last time we've looked at the water loss pieces, and we figured out that, well, one of the reasons why plants transpire that much water is because the ratio of water molecules to carbon dioxide in the atmosphere is such that for every 4 molecules of CO2 in the air, you have about 10,000 molecules of water that are present and therefore pouring out of the plant. So we as humans have it a little easier because there is a lot more oxygen in the air. So we also lose water by breathing, but not nearly as much as plants. So that's all fine and good. But now if we think about this, the water that has to get to the leaf to be transpired or lost, it ultimately comes from the soil. Well, granted, it comes with the big weather system, it comes down, infiltrates, ends into the soil, but somehow it has to get from the soil to the root, ascend in the whole system, and then to be transpired. Of course, the only way this works is if we maintain a gradient of sorts, right? Still, water has always got to go from high to low potential. So we can only extract water if we maintain a gradient in this direction. And there's some limits to this. One of the limits has to do with the leaf itself in upholding what we call Turgor pressure. Right? So I'll come over here so that you don't get lost. So if you think about this, all these are negative pressures, right? That's kind of concerning because it's basically a suction. You're pulling a slight vacuum on all the tissues here. And something makes them not collapse under that negative tension, under that pull. And the thing that does not make them collapse is the Turgor potential. And the way this works in a nutshell, let's use green, is that you have cells. And these cells have semi-permeable membranes. Meaning they exclude ions going one way, but they don't exclude water. Okay? And the way it works is that the plant accumulates ions in here. Okay? So here we build up a solute potential, Xi solute. And being that this is a semi-permeable membrane, solute potentials are always negative. Why are they negative? Well, they had that in a quiz question just recently. You have water being a dipole, so it has the electrostatic attraction that we've talked about in terms of cohesion and adhesion. Well, it also has that to any other ion that you add to the solution. So if you add sodium and chloride, or sodium chloride, for example, table salt, you have the positively charged sodium, the negatively charged chloride, and that will surround itself with that water cloud based on that electrostatic attraction. So it's always going to be a negative potential because to pull the water molecules away from these associations would take work. Okay, so now you have a negative potential here, and now any water on the outside of that cell would follow the concentration gradient to that more negative potential here. So if you have water at a lower solute concentration on the outside here, the water would tend to be wanting to go into this cell and thereby create a positive pressure within that cell. And that is what we call Turgor pressure. And that's how the plant stands upright rather than collapsing. So this builds Turgor pressure. So just to give you an idea of what we're talking about there in terms of potentials, if you have a 0.36 mole concentration of sucrose within that cell. Okay, granted there's more in it than just sucrose, but let's just go with sucrose. If you do the calculations there, the 0.36 mole of sucrose, to get how many grams of sucrose you have per liter, you have to multiply that by the atomic mass, which is 342 grams per mole for sucrose. So that turns out to be 124 grams of sucrose per liter of solution. Okay, and that 0.36 mole sucrose concentration of the solute here within the cell would correspond to a solute potential in the order of -1 megapascal. So this is how much sucrose you would have to put into the cell to maintain Turgor pressure to get to what -1 megapascal, which in this case, the leaf has already passed that, right? So if that was the Turgor pressure or the solute concentration within the cells surrounding the stomates and the leaf would be, this plant would not be happy, okay? So in other words, the plant actually has to increase the solute concentration within the cells to be able to maintain Turgor pressure so that the leaf water potential here can be set in such a way that you can still draw water up from the soil. So 124 grams of sucrose per liter, how much is that? Well, one of the beverages that maybe you have consumed in your lifetime. So that beverage has about 104 grams per sucrose per liter. Okay. So the concentration you have to get to -1 megapascals, right? That's more sugar than what is in the Coke. That's quite a remarkable amount of sugar you have to have in there. Okay. So that's part of the story. The other part of the story, of course, can be highlighted this one. So this is a pretty famous graph, and it shows potentials for the soil, for the root, that's the solid line, and for the leaf, which is the dashed line. And what they did here is they took a plant, an already established plant, and they just stopped watering it. And then they looked at these potentials as that changes over time. So looking at 7 days of that plant going to almost dying. So what you see is that as the water gets transpired by that established plant, the salt water potential decreases over time. So that's the top dashed line here. And it does so, of course, because the water content within that root zone is being depleted. Right? But it's a somewhat linear depletion, right? I mean, you see, okay, so it goes down from 0 megapascals down to, almost 1.5 megapascals. But what the plant experiences, of course, is what you see here in the root potentials and in the leaf potential. So on the first day of no watering, the root potential becomes more negative than the soil potential, and the leaf potential becomes even more negative than the root potential. So in other words, this is the day period, and this is the night period. In the night period, everything comes back together because there's no active transpiration. While there is active transpiration, you need to have these gradients to maintain the flow of water upward in the plant. Right? So that the soil and the root potential could be the same, or you wouldn't have any flux going on. But the lower the water content within the root zone becomes, the more pronounced these gradients become, right? So on the second day, the difference between the soil water and the root becomes much more in terms of the driving gradient than it was the day before. And same with the leaf. You just keep going like this until eventually you get to the point where the leaf potential would probably not recover at night anymore. So this is where the onset of wilting comes, to where at night it wouldn't come back together anymore. But now they would diverge, you would maintain some of those gradients, and now the leaf, where the solute concentration within the cells of the plant cannot be negative enough anymore to uphold Turgor pressure, and the plant wilts. So the water that is ascending to the plant is now at a potential that is in the order of the potential of the solute in the cells, and we can't uphold positive Turgor pressure anymore. We are wilting. The other thing that might happen is that the water within that column, so going from the root to the plant, goes under a high suction. That's not what you see in this graph necessarily. But that suction can only go to about 3 megapascals before the cohesion of the water in this long train no longer is enough to maintain a continuous column. And then you get something that's called cavitation, where the cohesive train of water gets disrupted and you get air bubbles forming in it to disrupt it. So that's part of that story. And now let's take another quick look at how this might all pull together. So for root water to happen, if you have a wet soil, then the idea is that you have relatively moderate gradients in that soil water potential. So the water that has to get from the bulk soil to the root only experiences moderate gradients, which also means that if the gradients are moderate, the hydraulic conductivity is relatively high. Versus if you get to a dry soil, you have really steep gradients around the root. So, and then with that drawdown of water in the immediate root vicinity, the hydraulic conductivity decreases significantly, and then you can't maintain fluxes that are high enough to meet the demand of that particular root there. So this is kind of the old way of looking at this. There's nothing wrong with this, but we've learned since that, well, roots and microbes are kind of helping with this a little bit. Okay? So if you take a closer look at the root, what we used to think is that, well, the water in the soil around the root becomes depleted. Thereby, you're starting to lose connectivity between the bulk soil and the root over time. But we've learned that actually there's a whole bunch of slimes that get exuded in that immediate root vicinity. It actually helps with the connectivity here. And that it is one of the explanations for why plants are actually able to withstand higher gradients here than we previously thought. So it's not just all physics, there's some biology there as well. So to kind of put this all together, well, we've looked at transpiration here on the left. So this is transpiration from a stomate. And on the right here, it is evaporation from a bare soil. And they're actually not all that dissimilar. Okay. So in both cases, we have somewhat of a cavity, right? I mean, the stomates, you got interior of the stomates. If you think about bare soil surfaces, you've got these kind of pores that are close to the surface. And in both cases, the water is lost from these air spaces, and the relative humidity of the air spaces, be it the stomate or the soil, is very, very high. Okay? With the stomates, pretty close to 100% all the time. With soil, pretty close to 100% relative humidity as well. It might decrease more towards 99, 98% when the soil is really, really dry. It has to be super, super dry to get to 98% relative. But not all dissimilar in a lot of ways. Okay? And then if we look at sort of the temporal evolution of water loss, they're also quite similar. Okay. So you might remember this piece here where it had the evaporative flux as a function of time, and it had this kind of behavior where we said, oh, there's a stage 1, there's a stage 2, and perhaps a stage 3 here as well. Where the stage 1 was, maybe I should have drawn this more as this horizontal line here, draw it like this. Where stage 1 was, the atmosphere is not limiting that flux at all. So this is, oh, sorry, the other way around. It is purely the atmosphere that is driving that flux of water. So the soil resupply of water is not limited. It's really just atmospheric demand in stage 1 that leads to that water loss. And in stage 2, we experience that soil limitation. Okay, so now the resupply of water to that open surface within the pores that are evaporating the water at the surface becomes limited. So the fluxes are no longer able to keep up with the atmospheric demand. And that's what stage 2 is. And then stage 3 is, well, the soil has dried out to the point where vapor transfer is the only thing bringing water vapor to the soil surface. So with transpiration, it's really not all that different. We could draw the exact same graph. I'll just call it T for transpiration as a function of time. Oh, wait, that's a problem. Let's call it trans, and this one T. And the behavior would be quite similar, if not the same. With one difference, there isn't really a stage 3, right? But there is a stage 1 and there is a stage 2. And the idea being the same. So in stage 1, soil is not limiting transpiration. Okay. So now here it's really atmospheric demand, how much energy is available to evaporate water, the wind, how much the surface resistance of the leaf and the immediate surrounding is. So that's what limits evaporation in this stage 1. And then as we get to stage 2, it is essentially the plant and the soil that are limiting transpiration. Okay. So water becomes limiting within the root zone. The first thing that's going to happen is that, well, the Turgor pressure is going to be affected, which means that the first thing that the plant will do is that the guard cells for the stomates experience that, and it will start to limit the water loss by closing the stomate. Right? So the transpirative flux goes down because stomates close. So that's number 1 of a reduction in water use by the plants. Closing the stomates always comes along with a yield penalty. Right? Those stomates can't take in carbon dioxide. So you're just going to be sitting there. You're not going to be growing. And what's worse is all your metabolic activities are still going on. So you're depleting your reserves of sugars while you're not being able to make new sugar. So you can't do that forever. You can do it for a little while. You can minimize your losses by partially closing the stomates. So the other part that then happens is that, well, that the flux from the bulk soil to the root, that slows down, right? I mean, the soil around the root becomes drier and drier. With that becoming drier, the hydraulic conductivity drops. So that flux of water to the root becomes really, really small and limits it that way. So this is soil root contact. Low fluxes. Okay. And I said there isn't really a stage 3 because if that just keeps going on, then we get to permanent wilting. There's no stage 3 in this one. You just drive down the transpiration and eventually the plant will no longer be able to do it. Okay. So not all that dissimilar between these two. And the other thing that's not dissimilar between the two is that this is an entirely passive process, right? I mean, the soil is the wick that connects soil in the subsurface with the atmosphere, and water just goes to it in accordance to these potential gradients. And the plant is the same way. Despite the fact that the plant is a living thing, it's an entirely passive process. It literally is like a big coffee string that you dip into a pool of water, and water just evaporates from that cotton wick all the time. So the plant does not use any ATP to make this happen. The water flows entirely passive through the process. Somewhere. There's one place where ATP is being used, and that is to accumulate the sugars in the cells, the plant has to use ATP. But for this transpiration flux, there's no energy needed, there's no biological activity needed. It really is just a straw or a paper. All right, so with that, let's figure out how one may do irrigation decisions to replenish the water in the soil to stay away from a yield penalty. So there's sort of two starting points to this. One is the one that we've already kind of discussed, and that is you just look at, for a given day, how much water evaporates from an open water surface. So we've looked at the average day in northern Idaho, the 5 millimeters per day evaporating. Right? So you could use that as an estimate to say, okay, so that tells me something about the atmospheric water loss based on this. The other one, and the probably more popular version, is to use a reference surface. And the reference surface is always a turf grass that is well watered, where the soil does not impose a limitation on the water use by that turf. And then you estimate the evaporative or the evapotranspiration from that particular turf. Okay? So it's reference surface or pan evaporation. If it is pan evaporation, then there's a few more steps. So you need to multiply that by some kind of pan constant. But once you do, that will get you either this way or that way to something that we call ET naught. Okay. So that is the reference evapotranspiration. So this is the reference ET, which is water loss due to atmospheric conditions. So that's the first step. You need to figure out how much water I need to put back on a plant for a given day. But so this one has no limitations of the soil. It doesn't say anything about the crop or plant yet at all. Okay, so in order to do that, we need a couple of multipliers. So the first one is a crop coefficient. Let me just make sure I get the nomenclature there right. Where's the page? Yes. So it's a KC, and that is a crop coefficient. And what's hidden in this crop coefficient? So it's going to be a number between 0 and 1 that scales that reference evapotranspiration. Is that every crop's going to be different, right? So some are better at evapotranspiring water with the full canopy. Some are not as good. And then the life stage of the plant matters as well, right? So if you've got just a teeny tiny plant, the transpiration from the teeny tiny plant is going to be a lot less than the reference evapotranspiration. Versus if the plant is fully developed canopy, it's going to be much closer to that reference evapotranspiration, right? So it's going to be life stage dependent and crop dependent, right? So if we got sort of two cases here, we got a teeny tiny plant, okay? So KC is going to be closer to, let's say, 0.1, versus you have a fully developed plant, closed canopy, then that crop coefficient is going to be closer to 1, plus the crop differences as well. So you can enter your life stage and crop type there. And then the next one is a multiplier for stress. And in this stress coefficient, you can have things like the soil water limitations. or salt stress, for example, too. So in the way this works for the most part, I'm just going to give you the example of soil water limitations. It all sort of maps back to the water retention characteristic. So you have a theta V HM. And you might remember the whole thing with the plant available water content, okay, that corresponded to 0.03 and 1.5 megapascals. So these are the limits of where your plant can survive. But if you think about the water uptake that would happen as you get closer to that permanent wilting point, stomates are starting to, close. The root water uptake just in the root vicinity is starting to limit it. So the stress coefficient here kind of accounts for how stressed the plant is as it gets to the extremes, and how the plant and soil system starts to limit the water uptake. So you just basically create yourself some kind of multiplier here that says that your coefficient of water uptake might behave something like this. Okay. So where this here is 1 and this is 0, and then you have a plateau here where you have no stress, which means that K stress equals to 1, versus if you get closer to permanent wilting point, then your stress coefficient reduces. The stress goes up, but the coefficient reduces, and the same as you get closer to saturation. So in other words, on this plateau here, you're transpiring at your maximum rate, so that the plant, takes full advantage of capturing carbon and losing water without any limitations, versus if you get closer to permanent wilting point, you're incurring a penalty. If you're going closer to fully wet situations, you're incurring a penalty. Okay. So you just multiply all this together, and what you get then is an estimated ET actual for a given crop. Okay. And that actual ET is the one that can inform your irrigation decisions. Right? So the way this works is that this reference surface ET is calculated with the famous equation called the Penman-Monteith equation. That kind of data is published by a variety of outfits. Here in the Pacific Northwest, it's Agrimet. They operate a whole bunch of weather stations and publish reference surface ET0 values. They also allow you access to crop coefficients, stress coefficients. And you can also find a whole bunch of, people have made cell phone apps or computer apps that will help you with this pull the data in and put these stress coefficients and crop coefficients in to give you an actual ET. And what the actual ET always is a hindsight estimate of how much water was lost the previous day, so that the farmer can make the decision to replenish what was lost yesterday with irrigation. Makes sense? All right, so that's, of course, part of the whole story. The other part of the story is that this is not the only way that you can figure out how to irrigate. So the ET estimates, that's part of this one here. So that's sort of a downstream estimate. You're doing it, the water has already been lost, but you're replenishing it. And in a lot of ways, some of the other things that inform that are the same way. So if you measure salt water content, it's the same thing. You're looking at the depletion of salt water content to react to it, to replenish the depletion. Same with canopy temperature, right? So if you put an infrared sensor on the plant, when you see the canopy temperature increase, that means the plant is stressed. It has already reduced its transpirated flux, right? And then you would be responding to that. Or even simpler, just looking at the plant and be like, oh, this looks a little wilted, I better water it. Right? So it's a downstream estimate. The damage has already, the yield penalty has already been incurred, you reacted to it. The holy grail, of course, is to do it as a forward-looking predictive thing, where you would say, oh, climate data tells me that tomorrow I'm going to lose this much water, so I'm going to put it on today to have it ready for tomorrow. So A lot of AI work going into this kind of work right now. And then the real-time one would be to use saltwater potential, because that tells you in real time what the plant is seeing. Right? I mean, that's as close to basically putting yourselves in the shoes of a plant and letting the plant tell you what the saltwater status is. So you can do all these things, and then I'll quickly flip through a couple of those figures here, which are actually quite obvious, and just get to this one, which kind of tells the story of, well, that's all fine and good, but then you also got to deal with variability within your fields. So, and then how do you incorporate the variability within your field into your irrigation and plant management decisions in general? That's the other part of the whole thing. This is a pretty typical picture on the Palouse, and as you get towards the end of summer, where the soil moisture storage patterns really come through, and where your grain ripens first is where you depleted the soil moisture, and then the greener areas where you still have soil moisture storage available, and the crop is not as far along in its life cycle as it is in the drier stages. Okay? So I'll leave it with